afternoon, everyone. And thanks for joining this great seminar series. So just a massive thank you to the organizers for putting together such a great event and allowing me to share some cool parasite pictures and how we use microscopy to help us understand the mode of action of antimalarials. So I'm located at the University of Pretoria, and I'm part of the Malaria Parasite Molecular Laboratory, also known as M2PL. We form part of an extensive national and international collaborative network with a main goal of eliminating malaria. But M2PL has three main research areas or main questions. So firstly is how the parasite functions, what are the important biological processes and the molecular drivers thereof. And then can we exploit these processes or molecular drivers in a drug discovery context? And then ultimately, can we use front-runner compounds for malaria elimination, as malaria is still a massive concern within Africa? So just a bit of background. So Plasmodium falciparum, which is responsible for most of the related deaths in Africa, has arguably one of the most complex and intricate life cycles in nature, which enables the parasite to survive within two different host environments, which has vastly different internal environments. So falciparum's life cycle starts off with a single sporozoid entering the bloodstream, moving to the liver and infecting these liver cells. Now, within the liver cell undergoes multiple rounds of asexual replication to produce thousands of merozoites, which enter the bloodstream, which then enter the bloodstream and then can subsequently infect red blood cells. So once inside the red blood cell, the parasite yet again undergoes multiple rounds of asexual replication to produce multiple new daughter merozoites, which then can reinfect red blood cells, and this process continues. But apart from that, this asexual proliferative stage, some of the parasites transition into sexual differentiation, where they transition through stage 1 to mature stage 5 gametocytes, forming mature male and female gametocytes, which then can be taken up by feeding Anopheles mosquito, and then once in the mosquito form male gametes and female gametes, which then fuse to form an oocyst, and within these oocysts produce thousands of new sporozoids, and this entire little cycle starts again. So in our lab, we firmly believe that it's essential to integrate fundamental biology with drug discovery, and knowing the mode of action or the primary target of an active compound can help us in our future decision-making and drug discovery efforts. So asexual proliferation is a key developmental process present during multiple phases of the parasite's life cycle, which harbors numerous proteins, which can then potentially be exploited as drug targets. But exactly how these processes work at the different life cycle stages and what the protein drivers thereof is not fully understood. So for the purpose of this talk, I will only be focusing on how the parasite is able to make new parasites within the asexual blood stage. So in mammalian cells, identical daughter cells are produced in a process known as mitosis. So during this process, the cell will duplicate its DNA. The DNA will condense into chromosomes. Microtubules will then originate from the microtubular organization centers. These microtubules will then bind to chromatids, pull them apart to different sides of the cell, ultimately forming two identical daughter cells. But now when we look at the mitotic process within the parasite, there are quite big differences between them, but there are also some similarities. This process is also known as zygogeny. So starting from a ring stage parasite, the parasite transitions into a trophozoid stage, which then subsequently enters zygogeny. And then within this process, the parasite undergoes multiple rounds of asynchronous DNA replication and segregation to produce a multinucleated parasite, or also known as a schizont, which can contain up to 32 individual nuclei. These nuclei are then packaged into individual merozoites. So This process is highly regulated by a number of proteins, but the master regulators are a variety of different protein kinase families. But I will mainly be focusing on aurora kinases. And these kinases are important for multiple steps during mitosis. And if any of their functions aren't performed correctly or something weird happens, the cells do enter a cancer-like state. And it's for this reason that they have been considered and explored as potential anti-cancer targets. And through the years, many aurora kinase specific anti-cancer compounds have been generated, some of them even reaching phase 2 clinical trials. So the parasite also encode for three aurora-like protein kinases that have been shown to be essential for this process from previous studies. However, their exact function during this cisogeny is not really clear. So we then asked the question, could we use known anti-cancer compounds to chemically probe parasite biology to gain a better understanding of how the mitotic process within the parasite functions or works? So first things first, we checked what effect the inhibitors will have on asexual proliferation. So here we just used a normal hemostained blood smear where one can evaluate numerous features like overall morphology, stage distribution, as well as basic parasite structure. So here I'm just showing you images of parasites taken every 12 hours within one asexual blood stage life cycle. So when ring stage parasites were treated with two different known anti-cancer inhibitors, namely ZM39 and asperidine, and left to progress through their asexual blood stage life cycle, we saw something quite interesting. These two inhibitors had a different phenotypic effect on the parasite, with parasites treated with ZM forming this enlarged food vacuole-like structure, while parasites treated with asperidine had a delay in asexual proliferation and halted in a mature schizont-like state. I was then also able to count the number of merozoites within these halted schizonts and compare that to untreated parasites. And I found that the schizonts treated with asperidine had significantly less merozoites present within them. So overall, it was quite interesting to see that two inhibitors, which should technically be inhibiting the same target or at least the same process, had two various or different phenotypic effects within asexual proliferation of the parasite. So effects on general morphology is good to know, but to really understand the mode of action of inhibitors, specific cellular structures need to be evaluated to answer these specific questions. So using structure-specific markers, dyes, and antibodies can help one answer these questions. Just for the remainder of the talk, I will be focusing on hesperidine as it has been characterized to some extent. So in previous work done by our lab, they evaluated how hesperidine specifically affects cisogeny. They used a cybergreen 1 DNA stain to stain the nuclear content of parasites, as well as an antibody specific to alpha-tubulin to determine if there is any effect on microtubule formation. And with the cybergreen, they wanted to see if the treatment affects nuclear segregation and nuclear morphology. So what they observed was that the nuclei of parasites treated with asperidine appeared abnormal and multi-lobed, and this subsequently led to there being less nuclei per schizont treated with asperidine compared to untreated parasites. Additionally, they saw when the parasites were treated with asperidine, they had a loss in alpha-tubulin signal, which indicates that asperidine treatment does affect microtubule formation. So these parasite images that were captured were quite of good quality, but some people might be thinking they don't look as good as mammalian fluorescent images. So Sabrina did touch on this. So this isn't this is in part due to this really small size of the parasite, which poses a challenge with resolution. So just to give you a rough estimate of how small the parasite is that we're talking about, here I compare the parasite to a Chinese hamster ovarian cell. And you can see it is quite small compared to the mammalian cell. So as Sabrina mentioned, we have this amazing sample prep technique called expansion microscopy, where you physically increase the size of your sample up to 5 times. So this, Sabrina and Ben made a really comprehensive and easy to follow protocol, which they were very generous, generous enough to share with us so we can try it out for ourselves. So I was able to go down to the AMI Imaging Center at UCT and use their expansion protocol along with the cool Zeiss LSM 980 with Eriscan 2. And lo and behold, this worked like a charm. We were able to expand the first ever malaria parasites in Africa. And we do see with great quality some cool structures like the microtubule organization centers, different microtubule structures like the hemispindle here, a mitotic spindle, as well as various other cool ultra structures. So I then used this expansion microscopy technique to evaluate what hesperidin-treated parasites look like. So on the left, you can see a mature schizont containing individual merozoites. Each little merozoid containing their rotary, two rotaries that Sabrina touched on, as well as the subpellicular microtubules extending from the apical ring. And you can see they are quite well divided and segregated from one another. But when we looked at hesperidin-treated parasites, it was quite evident that everything was bunched up. So we can see that the nuclear content was not really segregated that well, and the subpellicular microtubule signal was affected, where you can't really see where they start and end, and they look quite small compared to untreated parasites. So this just strengthened the previous data that was obtained, where hesperidin-treated parasites had abnormal nuclear segregation, a weird morphology, as well as the microtubules being affected. So this really strengthened the notion that we could use hesperidin as a tool compound to further study the mitotic process within the parasite. So what is next? We already touched on this. So we could ultimately form a nice phenotypic landscape where we could see, interestingly, 2 inhibitors of the same, that should be inhibiting the same process or protein having different phenotypic effects, and using expansion microscopy just to validate the effect that we saw with hesperidin-treated parasites, but just with a bit more or a lot of more resolution and seeing the ultrastructures thereof. So what is next for us? So we hope to combine the power of expansion microscopy, cell painting, and machine learning to generate morphological profiles to ultimately identify mode of actions of a compound with an unknown target of interest or have a novel killing phenotype. So this is being investigated by 2 amazing scientists in our research group, along with some collaborative efforts from H2D and UCT, as well as Anne Carpenter from the Broad Institute. So with that, I would just like to thank our group, the people that organized this cool seminar series, as well as our various collaborators all around the world, as well as the people that fund this cool research that we are doing. So thank you very much, and I'm open to any questions.
Henrico, thank you so much. That was an absolutely fascinating talk.